Hi students, how are you? Today we are going to understand the concept of chromatin. Let's begin. Your entire DNA, about 2 meters of it, is packed inside a nucleus that is just 6 micrometers wide. How does that even work? The answer is chromatin. And once you understand it, everything in cell biology starts making sense. Chromatin is simply a DNA protein complex found inside the nucleus. Think of it like this. Your DNA is a very, very long thread. You can't just stuff it loosely into a box. You need to wind it neatly onto spools. Those spools? They are histone proteins. The wound thread on a spool? That is the nucleosome. And a whole bag of spools? That is your chromosome. Chromatin has 2 protein components, histones, which are the most abundant, and non-histone proteins, which include structural proteins, polymerases, and gene-regulatory proteins. Now let's talk about histones. There are 5 types. The 4 core histones, H2A, H2B, H3, and H4, combine in equal ratios to form what is called the histone octamer. That simply means 8 histone proteins together, 2 of each type. They vary in their amino acid sequence, and they can be chemically modified through acetylation, methylation, and phosphorylation. These modifications are incredibly important because they change how tightly DNA is packed, and therefore which genes are switched on or off. This is the foundation of epigenetics. Then there is the 5th histone, H1. H1 is special. It does not form the octamer core. Instead, it sits on the outside of the nucleosome and helps compact the chromatin further into what we call the 30 nanometers fiber. There is exactly 1 H1 per nucleosome. So what exactly is a nucleosome? A nucleosome is the repeating building block of chromatin. The DNA molecule wins 1.65 times around the histone octamer core, using up 145 to 147 base pairs of DNA. The diameter of one nucleosome is 10 nanometers. Now, between each nucleosome there is a short stretch of free DNA called the linker DNA, typically about 35 nucleotide pairs long. The nucleosome core plus 1 linker region makes the nucleosome proper, which is roughly 200 nucleotide pairs in total. Look at the diagram here. Point to figure 1.19. This is exactly what it looks like under the electron microscope. A series of beads connected by a string. Each bead is one nucleosome. This is called the primary chromatin structure, or the beads on a string appearance. It forms a 10 nanometers fiber. Chromatin is rarely seen in this simple beads on a string form. It gets packed further. There are 3 key levels. Level 1, 10 nanometers fiber. The beads on a string. This is the primary structure. Level 2, 30 nanometers fiber. H1 histone folds the 10 nanometers fiber into a thicker, more compact structure. This is the secondary structure, and it is where most chromatin exists in a resting cell. Level 3. Loops and domains. The 30 nanometers fibers are coiled and looped into large domains. This is the tertiary structure. Individual domains can decondense and open up when a gene needs to be actively transcribed. How exactly does the 30 nanometers fiber form? Scientists have proposed 2 models. The solenoid model, nucleosomes are packed in a tight helical coil, like a spring, with about 6 nucleosomes per turn. The zigzag model. Nucleosomes alternate on 2 sides, with linker DNA crossing back and forth between them, creating a more open, irregular arrangement. Both models are still debated. The key point for your exam is that H1 is essential for this level of compaction, and it interacts with both the DNA and the protein components of the nucleosome. Now we come to the 2 main forms of chromatin in the nucleus, euchromatin and heterochromatin. Euchromatin is the open, loosely packed form. Under the microscope, it appears pale or light in stained sections, electron-lucent under electron microscope. It mainly consists of 30 nanometers fibers and loops. Most importantly, euchromatin contains the actively transcribed genes. A great example? Neurons. Nerve cells are highly active. They transcribe many genes, so their nuclei are predominantly euchromatic and appear pale and open. Heterochromatin is the opposite. It is the tightly packed, transcriptionally inactive form. Under the microscope it appears dark and dense. It is located mainly at the periphery of the nucleus, except over the nuclear pores, and adjacent to the nucleolus. There are two subtypes. 1. Constitutive heterochromatin, always inactive, in every cell type. This includes non-coding regions like centromeres. It never gets transcribed. 2. Facultative heterochromatin, inactive only in certain cells, during development or maturation. The classic example is the inactive X chromosome in females. Every female cell silences one of its two X chromosomes. This silenced chromosome condenses into a dark spot called the Barr body, visible near the nuclear periphery. In neutrophils, it appears as a drumstick extension of a nuclear lobe. Heterochromatin has specific methylation marks on its histones, which recruit special proteins that maintain the condensed, silent state. Let's quickly look at how chromatin appears in different real cells. Neurons, predominantly euchromatic, pale nucleus, very active transcription. Mature neutrophils, multilobed, densely staining nucleus, up to 90% heterochromatin, very few genes active. Orthochromatic erythroblasts, late red blood cell precursors, highly condensed nucleus, preparing to eject it completely. And the fascinating plasma cell, it actively produces a single immunoglobulin type, but most of its genome is silent. The condensed chromatin forms regular masses around the nuclear rim, giving it the classic clock-faced nucleus, a pattern you must know for histology. Finally, what happens during cell division? During mitosis, chromatin undergoes its most extreme compaction. Through multiple progressive levels of folding, 5 centimeters of chromosomal DNA is compacted 10,000-fold into a metaphase chromosome just 5 micrometers long. This is stabilized by protein complexes called condensins, which are essential for forming and maintaining the condensed mitotic chromosomes and ensuring accurate segregation into daughter cells. Remember the hierarchy: DNA, nucleosome, 30 nanometers fiber, loops, metaphase chromosome. So to summarize, chromatin is DNA packed with histones inside the nucleus. The nucleosome is the basic unit, 145 to 147 base pairs wound around an octamer core. Euchromatin is open and active, heterochromatin is closed and silent. And during mitosis, condensins pack it all into a compact chromosome. If this helped you, please like and subscribe to Gray's Gyan. Drop your questions in the comments below. I read every one of them.